The research paper that we will be discussing is on the regulation of gene expression with thyroid hormone in rats with myocardial infarction. In this study, a group of 12 weeks old female SD rats were chosen to conduct this experiment. The expression of hundreds of genes is altered in response to left ventricular remodeling following large transmural myocardial infarction, which involves changes in myocytes, interstitial matrix, and vasculature within the infarcted and non-infarcted areas. 55 genes were found to be altered with the treatment of thyroid hormone. Thyroid hormone improves left ventricular remodeling and cardiac performance after MI and has shown to reduce myocyte apoptosis. Thyroid hormone is also needed for cardiovascular development and homeostasis. Following MI, it is very common for patients to experience euthyroid sick syndrome due to low levels of thyroid. The diagram shows ligation, which is a procedure where a piece of thread is tied around the left ventricular coronary artery. This constricts the flow of blood, thus starving the heart of oxygen, which damages the heart muscle. Immediately following surgery, survivors were randomly assigned to different groups, which were sham MI, MI, and MI plus T4. T4 pellets were implanted subcutaneously in the MI plus T4 animals shortly after induced myocardial infarction. Both sham MI and MI were implanted with placebo pellets at the same time after surgery. After 4 weeks of treatment, total RNA was isolated from LV non-infarcted areas for analysis. The study was conducted with several procedures and measurements. The ones that we will be analyzing are real-time PCR, microarray, Western blotting, and densitometry.
Signals were detected in 13,188 genes out of 22,523, of which the expression of 154 genes were decreased and the expression of 200 genes were increased, but we will only be discussing a few of the genes. The table above shows results from the microarray analysis, which allows us to measure the gene expression levels. There were 6 genes downregulated by MI but upregulated by T4 treatment. This can be seen in CARD9 gene, where the expression level in MI is 0.63, which is a decrease, whereas with T4 treatment it increases to 0.85. And there were 12 genes upregulated by MI, but downregulated by T4 treatment. If you take a look at ANGPT2 gene, the expression level in MI is 1.21, which is an increase, and again with T4 treatment it decreases to 1.01. This table shows the comparison between microarray and a real-time PCR of the following 5 genes: COLAA1, GPX3, NPPA, IFI27L, and beta-MHC. Real-time PCR is a technique which allows to amplify and simultaneously quantify a targeted DNA molecule and is more sensitive than microarray. The fold changes in the expression level of targeted genes revealed by RT-PCR were similar to or higher than those determined by microarray. The table here shows that with T4 treatment, expression levels changed. For example, in the gene beta-MHC, the microarray did not show much of a difference, but the real-time PCR showed a decrease in beta-MHC expression with T4 treatment, going from 1.80 to 0.83. Western blotting is used to identify a specific protein in a given tissue sample, and protein levels are evaluated through densitometry. Angiopoietin 2 expression was increased in non-infarcted myocardium after MI decreased with T4 treatment. This shows consistency with Figure 1 microarray results. The graph shows the density of angiopoietin in the Western blotting, and by looking at it you can see that there is much more angiopoietin in both sham and MI than T4, which supports that the gene expression decreases with T4 treatment.
Microarray results illustrate that more genes in the non-infarcted myocardium were detected with altered expression at 4 weeks after MI. Microarray results show that T4 treatment decreased the expression of 27 genes and increased the expression of 28 after MI. It also showed no difference in beta-MHC expression in the non-infarcted myocardium at 4 weeks after MI with or without T4 treatment. RT-PCR technique showed a tendency of increased beta-MHC gene expression after MI and a decrease in beta-MHC expression with T4 treatment, but did not reach statistical significance. When compared, the selected genes examined both microarray and RT-PCR. RT-PCR has presented a high level of change in expression when compared to microarray. Therefore, this demonstrates that RT-PCR is more sensitive than microarray and the Illumina microarray platform that has been used. The recent study only focused on the cardiac gene expression changes at 4 weeks post-MI. However, the post-MI LV remodeling was continued for even longer than that. There may have been more differences in cardiac gene expression. The observed effects on cardiac gene expression following T4 treatment might be partially due to its effects on the heart rate and peripheral vascular resistance, which cannot be excluded by the present study. Other molecular mechanisms involved in thyroid hormone effects cannot be underestimated, such as non-genomic actions and changes in microRNA expression. Overall results shows that thyroid hormone can reduce myocyte apoptosis. This can be proven from this results shown. For example, in figure 3, it clearly shows that there is a relationship between angioprotein 2 and myocardial infarction. The different results which have been conducted also proves that altered expression of genes might have been involved in the beneficial effects of thyroid hormone treatment following MI in rats.